I had I remember I was like,
all right. Did I miss anybody who are giving that their exam?
they are, some of them can be extremely difficult, so I had to like train up what. Okay, I can picture, but that's not fun. Yeah. There are messages that are, I don't know how to describe them, but they are, no. Like hot stairs. No, they're not words. Otherwise, I would never would be the exact. My opinion. Every time I go to the doctor, they're like, I asked this weekend, don't go to that's why. Because there's like, they're not real dogs. But I also got a knife. And there's like, so. Statistically significant amount of people that are.
All right, did I miss anybody else?
I was like, you have to go to like whatever. He was like,
All right, let's go ahead and start class today then. Hopefully I watched that video I posted on Tuesday. I'm feeling much better. You might notice my voice is still a little gravelly. You heard my second lecture of the day, so I apologize if I have to like take a break every now and then. Hopefully you did watch that lecture. We will review the questions today in class. Let's see, I passed back exams. There is opportunity to get points back, all right? First of all, if you got anything wrong on fill in the blank number 5, that's the one asking about monomers and polymers. The one where I made a mistake, I did not catch my mistake when I was editing it. I think I said it was a new nucleotide was the polymer name. It should have been nucleic acid. You got that question wrong at all. I did not take off any points because I felt that was unfair. There are opportunities to get some more points back. You can get the full points back for short answer 9C. That is the one with the polar bears and the alligators. I really like this question because there's so many different layers to how you remember what you study here. And I think a lot of you understood the content, but maybe didn't quite understand what I was asking. So I'm giving you the opportunity to get full points back on that if you go in and correct it. I've given you a hint on the full instructions for this exam back opportunity that's linked on Moodle. But essentially both the polar bear and the alligator should have similar membrane fluidities, right? And I want you to tell me what their membranes might be composed of, because one lives in a cold environment and one lives in a hot environment, right? How are their membranes different to obtain that similar fluidity between the two? And then I have 5 questions here that I've given you an opportunity to get partial points back, right? So these were where a lot of you got them wrong. It wasn't necessarily the majority of the class that got it wrong, but enough of you that I felt that maybe I might have worded something unfairly. There was a disconnect somewhere. If you correct those answers, if you got any of them wrong, I'll give you half points back on those. So in order to do this opportunity here, what I am asking is for you not to erase the incorrect answer on your exam, but rather write the correct answer next to it, or if there's no room, use a separate piece of paper. And then on a separate piece of paper, give me like a 1-sentence rationale as to why your answer is now correct. Next Thursday, which is the next time you will meet as a class, since Nobel Conference is next Tuesday, I will have you turn in the exam with your rationale sheet, and I will go through those corrections. If you turn your exam back into me, I will get it back to you eventually, right? These exams are for you to keep. I'm not going to collect them at the end of class. Any questions? All right, so you'll only be able to correct those questions that I listed up on the board. There is a reason for that, right? I have based these questions on statistically how many, what percentage of you got that wrong, and that's how I made my determination as to which questions I would give you the opportunity to points back. All right. I had to watch that video before class today, and I actually ended class class before the exam on this review question. So the first part of class is us going to be working through a series of questions to make sure that we understand the content and to give you a chance to ask me questions if you do. So we're going to start with this question here, having think all the way back to energy here. You might have to review your notes, talk with your neighbor, but I'll give you a couple minutes to answer this question.
I don't even know what this is.
But that's not what we're taking.
Oh. Okay.
Extra segments to hand back to you.
I mean, you want to like it. I was going to ask. I think it has to do with energy going in and energy going out. Exergonic is energy out. And the only one that would qualify is exergonic. Like net energy going out. Yeah, it's okay. It's producing more energy. Yeah. Okay, so energonic requires energy.
All right, I need two graphs up here. They have different values for the change in free energy. Which one is going to happen spontaneously? Is it going to be the one on my right or on my left? On my right. Yeah, because that delta G is less than 0. That should be the biggest hint for you there. Right? If it's less than 0, it will happen spontaneously. It does not need an energy input. All right. The second question here is a reaction that has a delta G of 130.4. All right, so it's an endergonic reaction. We're going to couple it with an exergonic reaction. Which of these 4 reactions with different types of energy would have to be coupled with the endergonic reaction to make it spontaneous? A, yeah, because we need to get this delta G, its change in free energy, less than 0. The only one that we could add with 130.4 is negative 394.36 in order to get a negative value for that change in free energy. All right, any questions here? All right, we're going to go to our next one here. We started talking about enzymes in the video. I want you to answer this questions about what would happen if enzyme C became non-functional.
Yeah, it was so they can't make a problem. Yeah, we know. Yeah. It depends on the reaction. It would be broken down either by a different mechanism. Sometimes it would be broken down, sometimes it would just have like the one. Because I've heard that for alcohol, if someone has like a less functional enzyme in there, intermediate product. Speaking of alcohol, have you heard of auto breweries? Yes. Isn't that so? I know. You can just they're like, yeah, this is gonna be here. We made a study report. Now that it's become like more of a mainstream. I mean, it's easy stuff because carbs without alcohol is your big head. Yeah, alcohol. But yeah, I have negative feelings about alcohol. You don't like it? I don't like its taste loud. Like, I can't stand like alcohol percentage of like 3%. I can't stand any of it. I can, well, okay. Part of the reason why I don't like alcohol is this medicine, this herbal medicine that I take.
All right, enzyme C has become non-functional. Would this affect the rate at which this entire reaction occurs? Yes. How would it affect the rate?
The enzymes are like very specific to their substrate, so if there's no enzyme C that's not working, there's not going to be another enzyme that's going to fit the substrate, which is going to cut like overall like the entire function of what it's making.
Yeah. So the entire function of this enzyme, if it's no longer working, it might cause this entire reaction to stop, right? Or there's another possibility. Did someone come up with a different answer? There is a way that maybe it won't stop, but it would drastically slow down, right? If this reaction happens to be an exergonic reaction and happens spontaneously, right? So it doesn't need an enzyme per se. It could still go, but without an enzyme, it's going to be much slower. So either it's going to be much slower or it's going to absolutely stop, right? Depending on what exactly that reaction is. All right, so what does that mean about the production of the product being made? Is it going to be made?
No, or very little.
No or very little, yeah. Because enzyme C is basically put a stop to that product being made, or it's very much decreased the rate at which it's being made. What about, let's look at substrate 3 here. What's going to happen to substrate 3? Yeah, it's going to start accumulating, right? Because substrate 3 will no longer be able to be used by enzyme C because it's non-functional. We'll probably get a lot of substrate 3 start accumulating, right? Either it's going to accumulate or it's going to accumulate and slowly deplete, right? If the reaction can happen spontaneously. So, a change in the enzyme function can drastically change the overall reaction. Any questions here? Yeah?
Is this what happens to the intermediate breakdown product of alcohol when someone has like issues with their alcohol like enzymes?
Ooh, very likely. Yeah. I would say that's probably a good hypothesis as to what's happening. All right, let's go to the next question. I asked you about catabolic and anabolic reactions. I want you to tell me what type of reactions you're seeing on the screen.
Yeah, so anabolic means right? Oh, it is catabolic. Yeah. I just think like A is the base or not. Right. Not.
I think the first sections will be interesting.
Versus a catalyst for energy.
We
have to go.
This class will have to go at least 2 talks.
What?
Yes. You can go in person or virtual.
Okay. Is there like a
Yeah, it's like 5 minutes.
real? Okay,
wait, you have to email them.
They're conditioning viewers.
Out of what? It might be a 3 or 4.
0 out of 5, and that's a really good deal.
Is that a Nobel? Yeah. It's like interesting.
Did you get any water bottle? It's so cute. I don't care. Oh. I pulled out a bunch of signs. I'm not gonna get any. It's actually not a good thing. I'm really bad too.
We want you to go to Nobel. I was such a cool Yes.
It's fine. They put it on.
All right, I have 2 reactions up on here. One reaction, we're taking cellulose and breaking it down into glucose. Another reaction, we are looking at some nucleotides that are being combined to create a nucleic acid. Which one of these is a anabolic reaction? The cellulose or the nucleotide? The nucleotide, yeah. This one over here, right? This one is the anabolic reaction because we're building up a larger molecule. We're taking smaller components to build up a larger molecule. All right? So cellulose breaking down into glucose, what type of reaction is that? That's the opposite one, right? It's catabolic, right? So I have catabolic and anabolic reaction up on the screen. Catabolic, we're taking a larger molecule and we're breaking it down to its smaller components, right? Now I like to remember it like catabolic is like cannibalism, right? We're breaking something down. That's how my mind works. Maybe your mind doesn't work like that, but that's how I remember it, right? And then anabolic is we're building something up here. Any questions? All right, we'll move on to our next one. All right, in the video we talked about these molecules that provide energy for a cell. I want you to tell me, how do these molecules provide energy to the cell?
When they release energy. Yeah. NADH is more like amines to get ATP. So it's like it's less valuable. ATP is more direct. Yeah, ATP it's frying phosphate turning into diphosphate. Yeah.
Well, if it's $2, it's 7 days. But that's not that's like not that much, but I was like,
Okay, good.
I think we're doing it. We usually do. I think it's the second one.
I know, if you would have said what type.
All right, we have ATP, we have NADH. We talked about it in that recorded lecture. They each have a way to provide energy for the cell. How does ATP provide the energy? Where does that energy come from?
Phosphate bonds.
Yeah, energy is stored in the bonds of those phosphate groups. See if I can get my answer. There it goes. Right, so those phosphate groups on ATP hold a lot of energy. And when those bonds are broken, that energy is released, right? And that energy can then be harnessed by the cell to perform whatever process it needs energy for. Right? So we're holding the energy within the bonds. What about NADH? Yeah, it's a carrier molecule. It can carry electrons, right? So it provides energy by the removal and the acceptance of electrons and protons. So it can transfer energy, right? So it can pick up some electrons, it can give away electrons, right? And in that process, it can transfer the energy associated with those electrons. All right, thinking back to last unit, what organelle plays a huge role in providing energy for the cell? The mitochondria. You've probably all seen the meme, right? Powerhouse of the cell because it produces a lot of ATP and NADH. All right, any questions here? All right, our next question. And for those of you in chemistry, I understand that you've gone in much more detail on redox reactions than this. But I want you to answer this question. Which of the products, or excuse me, which of the reactants are being oxidized and which are being reduced? Those charges. I have not made it obvious. Yeah.
If it's gaining an electron in the number that's
Yeah, so I'll be reactants because I
Honestly, I don't know. That was, I like the one that I think it was.
Oh, wait.
I know.
I'm totally red velvet or cheese.
the door.
CR plus, unless it was like negative.
Yeah. No. Yeah, because the gain is less than CR. No. Because it loses one, so it makes it more positive.
We know what we're doing. Sort of.
So it's more positive. Those are, that's not the electrons, that's the Such it. So like this, CR on the left has 1 plus, and CR on the right has 2. So the CR be. Yeah. It's like opposite, so if there's electron, you get more positive.
All right, of these atoms, which one is being oxidized? Did you say chromium? Yeah. Chromium is being oxidized, and SN, which I cannot remember the name of the full, do we know what it is? Is it tin? All right, tin. SN is being reduced. All right, let's talk about why, because for some of you, maybe this stumped you. All right, so if we look at chromium here, it has a positive charge, which means that it is an ion, and it is an ion that has a 1 less electron than it does a proton. Right, so it has 1 more positive charge than it has a negative charge. Hence the positive charge. And we're going from chromium positive to chromium 3 positive, right, which means it's losing even more electrons in order to get an even more positive charge. Now it has 3 more protons than it has electrons. All right, opposite being the case for the tin, right, we're going from SN4 positive to SN2 positive. So SN4 positive means it has 4 more protons than electrons, giving it that 4 positive charge. And it's going to a 2 positive, which means it's actually gaining electrons, right? It's gaining 2 electrons. So now it only has 2 more protons than it has electrons. Right? So it's gaining electrons, it is being reduced. So I like this allow me know what you call it, like mnemonic of oil rig. I know there's also what is it, Leo goes grr, Leo the lion goes grr. Losing electrons is oxidation, gaining electrons is reduction. So there's a couple of ways that help you remember this. Any questions here? All right. Our last review slide before we move on to more lecture here. We started talking about cellular respiration, and we started talking about that first step in cellular respiration. I want you to fill in the blanks. What are the intermediate molecules that are missing in these blanks on this diagram? And I'll give you a few minutes. And while you all think, I'm going to go refill my water bottle.
But I like it better today. It's on top of
Yes, it's like the molecule that's like
You're better either.
I started grabbing
All right, we talked about glycolysis and the 3 stages of glycolysis. But I want you to fill in the blanks with the molecules that are like the intermediates or the end product of these stages. So this first one, we're taking glucose, right? And we're splitting it into 2 different molecules. Or excuse me, before we even split it, we're turning it into a different molecule, all right? So what would that molecule be called? Yeah, fructose 1,6-biphosphate. Right, that's the first intermediate molecule. Yes, these are the molecules I would expect you to know on an exam. All right, so glucose is being changed into fructose 1,6-biphosphate in the energy investment stage. All right, so we're using some ATP here. Now we're going to cleave this fructose 1,6-biphosphate into 2 molecules, right? And what is the name of those 2 molecules? Glyceraldehyde 3-phosphate, yes. Now sometimes this is abbreviated as G3P. If you use that abbreviation, I would be okay with it. But I'm going to put the caveat here that when we talk about photosynthesis, we're going to talk about 3GP. So 3GP and G3P can get really confusing really fast. I would recommend you say the full name just to avoid that confusion. All right, so glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is going to continue on this process in the energy. We're producing energy in this stage here, and we're getting our end product. What is the end product's name here? Pyruvate. Yeah, we're getting pyruvate. And where does this process occur in the cell? In the cytosol, yeah. So we're not quite in the mitochondria yet, right? We're in the cytosol, but we're about to move into the mitochondria. Any questions about glycolysis? All right, so as you're studying, these are the terms I would expect you to know, as well as the names of the stages here. Now I know it's a lot of terms, but it's actually very much simplified from what I could be making you know. So I have just cherry-picked the terms that I felt most important in this process. All right, so let's continue on with cellular respiration then. Glycolysis is the first step, right? But let's move on to talk about the next step. So in glycolysis, we've made pyruvate, right? 2 pyruvate molecules per 1 molecule of glucose. Those pyruvate molecules are going to enter into this next stage, which is the breakdown of pyruvate. And notice this next stage here, this is happening inside of the mitochondria, right? We're moving pyruvate inside of the mitochondria. All right, so breakdown of pyruvate. The pyruvate is being moved inside of the mitochondria, and we can see that it's being moved via these channels and these symporters. Right, and that should make sense because if we look at the molecule of pyruvate, It's relatively on the larger side. It has a lot of oxygens, and it's even denoted on the molecule itself that there's a negative charge associated with one of the oxygens. So it is a polar molecule, right? And we know that polar molecules do not just simply diffuse through the plasma membrane. They need a protein to help them along. And so that is exactly what is happening here. Once it's inside, once it's inside of the mitochondria, a couple of things are going to happen, right? The first thing that's going to happen is that we are going to combine our pyruvate with acetyl-CoA, right? Or with CoA in order to make acetyl-CoA. So pyruvate is going to be combined with this molecule called coenzyme A. Coenzyme A with pyruvate is then going to make acetyl-CoA. Right? So that is kind of like the end product of this step is the acetyl-CoA. But in the process of turning pyruvate into acetyl-CoA, we're also going to produce some CO2 as well as a molecule of NADH. All right, so pyruvate is changing into acetyl-CoA. Right? We're adding in the coenzyme A molecule to make this happen. We're also going to be using an enzyme to help aid this process. Right? And that enzyme is pyruvate dehydrogenase. Right? That helps put the coenzyme A onto the pyruvate molecule. Right? And as well during this process, we're removing some CO2. CO2 not really being the purpose of cellular respiration, but it is a product being produced. It's kind of like a byproduct. It's not why we go through cellular respiration, but it's produced through cellular respiration. And we're also getting NADH. All right, so we're yielding one NADH for each pyruvate molecule. But remember, a glucose molecule makes two pyruvate molecules. So per glucose molecule, we would be getting two NADHs. Right? Keep that in mind. That's something I'm going to be reiterating as we go through these steps. Right? What happens during this step, and then what happens per glucose molecule. All right, my next slide is basically just streamlined this entire process. All right, so thinking big picture here. CO2 is being removed. We're producing NADH. All right, we're also getting an excess of a hydrogen ion as well. We're attaching the coenzyme A to acetate, which is the intermediate of this pyruvate molecule. Notice that acetate is not bolded. It's not something I would expect you to remember. Okay? But the end result here is an acetyl-CoA. We have an acetyl-CoA that is being produced and is ready to move on to the next step.
When you say he wants to remember the specific molecule, he wants to draw it to?
The name. I wouldn't expect you to draw it. The name, what stage it occurs, when it occurs. Yeah. Here's already enough for you to learn. All right, let's review these first two stages. I'm going to have you fill out this table, and we will continue to add on to this table as we keep talking about the next couple of stages. But I'll give you a couple minutes to answer, where does this occur, what is the end product, and how many energy containing molecules per one molecule of glucose is produced. Yeah, so in the table I'm asking you in general what is being used. And then just asking for a specific number of molecules. What is that product of the same? excited about for the Macomb conference?
I'm excited for the pastry show. I think that's gonna be fun.
I think so too.
Yeah.
And I think they're giving their talk in French, because I know they have a translator.
Oh, that's fun. That feels a lot more like authentic,
I
right?
know. I'm excited for that one too.
Yeah.
I think there's one like the history of sugar, which I think will be very interesting.
Yeah, I mean, I knew it's kind of heavy.
Yeah, it'll be a little heavy.
Yeah,
but
but
yeah.
cool.
Yeah.
Yeah. Should be a good group.
Yes.
I don't know. I hope not. I would visualize.
No, it's just that there's a lot of racism, history of racism and things like that
It's kind of sideways.
enslaving not just black people, but also
All right, so let's fill in this table. Glycolysis, where does this stage occur in the cell? We kind of already answered this one. In the cytosol, yes. And what is the end product of this stage? Pyruvate, yeah. In fact, per glucose molecule, it would be 2 pyruvate.
You
Yeah,
have to know like how much ATP and NADH it makes.
I would know, yeah.
How much is
Well, that's a great question. For glycolysis, what is the net yield of energy-containing molecules? 2 ATP, yes. All right, so per molecule of glucose, we're going to get 2 ATP. But let's not forget about the NADHs. There's also going to be 2 NADHs per glucose molecule. All right, so we have glycolysis. Let's move on to the breakdown of pyruvate. Where does this stage occur?
Mitochondria.
In the mitochondria. What is the end product of this stage? Acetyl-CoA. And what is the yield of energy-containing molecules? I hear NADH. How many NADHs? 2, because we're talking per glucose molecule, which means each pyruvate, remember there's 2 per glucose molecule, goes through this process twice. Right? Each time the process happens, it's 1 NADHs. We're doing 2, so we have 2 NADHs. All right. We're going to keep moving on here, and we will keep filling out this table as we go. So let's talk about that third stage. The third stage is the citric acid cycle. Right? Now this stage is a little bit different in that it is a cycle. Right? There's not really a defined end product. Right? If we're thinking about glycolysis, the end product is pyruvate. But in this case, the end product is something that just immediately goes back into the cycle. All right, so it is a metabolic cycle. We're going to look at a few key molecules. We're going to look at some molecules that you always will hear when you hear the citric acid cycle. We're also going to see what is being produced throughout the cycle. Now, I'm not going to ask you to memorize every single intermediate molecule. We're going to focus on the big ones here. All right, so let's jump into the citric acid cycle. All right, citric acid cycle. We have acetyl-CoA, right? That is the end product of the breakdown of pyruvate. Acetyl-CoA is going to enter into the citric acid cycle. And we see that over here. Now, before acetyl-A enters into the citric acid cycle, it's going to lose its CoA, right? So coenzyme A is going to go away. And what's left, right, this acetyl group, will combine with oxaloacetate. So that is the first molecule name I want you to know of the citric acid cycle. So the acetyl combines with oxaloacetate, and once it combines with oxaloacetate, it becomes citrate. Hence the name of the cycle, the citric acid cycle. All right, so acetyl-CoA is entering into the equation. It's dropping the CoA bit, and the acetyl bit is going to combine with oxaloacetate. And oxaloacetate, when it's combined with acetyl, it's going to become citrate. All right, what happens to citrate then is going to be a series of reactions. Right? Citrate is going to turn into another intermediate molecule. that intermediate molecule is going to turn into another intermediate molecule, and so on, right? There's many different steps. In fact, it's labeled here. There's 8 different steps. I'm not going to have you know any of the names of the other molecules or steps, right? I only want you to know oxaloacetate and citrate. All right, but as citrate moves through the cycle and becomes an intermediate that becomes another intermediate and so on, we're going to see the production of some energy-containing molecules, right? So first of all, we're going to get some CO2 produced. So this is not an energy-containing molecule, but rather a byproduct. CO2 will be produced. We're also going to get 1 ATP being produced, as well as 3 NADHs. Now there's one more molecule being produced, and that is this molecule of FADH2. We're producing one of these per turn of this cycle. Now I haven't really introduced FADH2, and I'm not going to formally introduce it, but what FADH2 is it's a carrier molecule, right? It functions very similar to NADH, right? It carries electrons. All right, so we're producing some CO2, 2 CO2 to be exact. We're producing 1 ATP, 3 NADHs, and 1 FADH2. This is per 1 turn of our cycle, per 1 molecule of acetyl-CoA. Right, now if we think back in terms of per glucose molecule, how many molecules of these ATP, FADH2, NADHs are going to be produced per glucose molecule? Let's look at just ATP. How many ATP would be produced per glucose molecule? It would be 2. Yeah, because remember, a glucose molecule gets cleaved in half in glycolysis to become pyruvate, 2-pyruvate. Those 2-pyruvate become 2 acetyl-CoAs, which means we have to run this cycle twice in order to account for the 2 acetyl-CoAs that happen per 1 glucose molecule. All right, so keep that in mind. All right. Now I mentioned it's a cycle, right? So there's really not a defined end product because the products just keep being regenerated and reused and the cycle continues. One of the biggest steps of regeneration, one that's very important, is the regeneration of oxaloacetate. All right, so we have this acetyl group. It combined with oxaloacetate to become citrate. And that citrate's going to become a variety of other intermediate molecules. Right? We've essentially used the oxaloacetate. Now, in order for us to process another molecule of acetyl-CoA, we have to regenerate that oxaloacetate. We have to make it available again. And that's what happens in this cycle, right? Citrate is going to become a variety of other intermediates until finally it's this molecule malate, and malate is going to turn into oxaloacetate. Right, so now we have oxaloacetate back, we can continue the cycle. We can process another molecule of acetyl-CoA. Now it just so happens within that process, that's where we're getting one of our molecules of NADH. Right, NADH is being produced when malate turns into oxaloacetate. Right, so it's a cycle. Oxaloacetate will be used for the next acetyl-CoA. It'll be regenerated, and then be used for the next acetyl-CoA, and then be regenerated. All right, so citric acid cycle. It's a series of reactions. I'm not having you memorize the whole thing. I don't think that's pertinent for a Bio 110 class. I want you to understand why it happens, what is being produced, and the major players. Now, if you take another class, maybe biochemistry, then it might be more appropriate that you would have to learn all these intermediates because you might be talking about the reactions a little bit more in depth. All right, so citric acid cycle is now completing the oxidation of the organic molecule, right? So we started with glucose, our organic molecule. It has been completely oxidized at this point. And in the process, we have created a lot of NADH and FADH2, right? Now remember, per 1 glucose molecule, some of these steps are going to happen twice. right? So glucose splits into 2 pyruvate. So the breakdown of pyruvate happens twice per glucose molecule. Pyruvate becomes acetyl-CoA, right? So that happens twice. I think I said the breakdown of pyruvate for that previous step. But pyruvate is going to be broken down into acetyl-CoA twice, right? So this is per 1 glucose molecule. Some steps happen twice. So thus the citric acid cycle, after 2 turns, we have 4 CO2, 2 ATP, right? So we have some ATP, which is great. We want ATP. But we generated a lot of NADH, right? 6 molecules of NADH, as well as a couple of molecules of FADH2, right? So we have a lot of molecules of these energy carriers. Right? And this is going to become important when we talk about the last and final step. Right, so this is just the citric acid cycle at this point. Now remember, we've also created NADH in glycolysis, as well as the breakdown of pyruvate. Right, so there's more than what's just listed on this screen. All right, I want you to fill out the table again, and if you get done early, you can find a mitochondrial disease that you might want to share, because there are many diseases that affect the mitochondria, and as you can imagine, mitochondria producing energy, this can be a big deal. I'll give you a few minutes.
How did we figure all this out?
Lots of research, lots of experiments.
Okay.
Yeah. And that's a lot of biochemistry, I'm sure. I don't even know like the big historical player. I know like Krebs came up with the citric acid cycle, because sometimes the citric acid cycle is called the Krebs cycle.
Oh, yeah, okay.
Yeah. So I know he's a major player, but I don't know the others. Many years of research.
This is anaerobic respiration, right?
This is aerobic. So this is where we need oxygen.
Okay.
But some of these steps could occur in anaerobic. Like glycolysis can still occur in anaerobic.
Okay. What is, like, if you say the Krebs cycle, that sounds familiar.
Yeah.
Because, like, what is produced in the anaerobic respiration again that causes, like, muscle pain? What is that thing that?
I think we're thinking fermentation. So fermentation is when only glycolysis happens, and we get a buildup of lactic acid.
Okay.
That's what causes muscle cramps. Yep. Anaerobic respiration, and we'll talk about that in another lecture, is actually all of these steps we're going to talk about, except it doesn't use oxygen, it just uses a different molecule in its place.
But, okay.
Yeah.
It's just a lot less effective.
Yeah.
I think the hardest part here is to not get them confused.
Yes.
Oh, so.
Yeah. It's a shorter unit, but it has a lot of detail in this unit.
Which is probably why before the next exam, that's
Or
why
did-
we're going to use one side by side and be
Yes.
or the day. Here's where that is different.
Repetition in studying is going to be key for this exam.
Thank you. Yeah, this is an AI, and that, so what we're doing with AI, that's what we're broken down.
And it makes another 2 NADH. Then what do you think? Not like reading the first before NADH per molecule, or is there no NADH?
Then don't use it. Which is what I did. Well, this is my idea. This is my idea. I just told you. This is my
I know it's 2,
Because
but
I told you.
is it 2 plus 2, or is it?
Yeah, see? I did that there. But yeah, this is my idea, which is why I'm
NADHs, right? Or not?
In which step?
In the first step.
Okay.
Okay. And then in pyruvate it makes another 2 NADHs, or is it the same? Okay, so then in total it needs 4 NADHs for one.
Yes, I know.
Okay.
Yes.
Okay.
Yes. Okay. Because when we're talking about glycolysis, we're talking about one glucose molecule.
Right.
Even though it's being broken.
Yes.
And then when we start talking about breakdown, pyruvate, and citric acid cycle, now we're talking about like 1/2 of glucose at a time.
Okay.
Yeah. All right. Citric acid cycle, where does this occur? It occurs in the mitochondria. Yes, we are still in the mitochondria. What is the end product of this stage? This is a little tricky because I said there really is no end product. But what would you consider an end product here? What's the last step of the cycle? I would say that is part of an end product, but maybe an energy-containing end product. I would say the end product here is actually like oxaloacetate being regenerated. It's the last step of the cycle, but it's not really an end product because it's just going to be continually used. Right? If we're thinking about glycolysis, the end product is pyruvate. We know that. Right? But this is a cycle, so it gets a little muddy. We're in a little bit of a gray area here. But there are other products being produced, right, as a result of oxaloacetate being regenerated and going through the citric acid cycle. What is the, let me ask you about the number of ATPs. How many ATPs are being produced in the citric acid cycle per 1 molecule of glucose? Yeah, 2. 2 ATP, 6 NADHs, 2 FADH2s. I also included the CO2, which is not an energy-carrying molecule, but I wanted to put it up there because it is being produced. All right. Did anybody research a mitochondrial disease that they want to share? I don't have one at the top of my head. That's okay. My last class didn't research one either, so we'll just skip over this one. All right. Any questions before we move on to the last step? All right, so far, the culmination of the first 3 steps of cellular respiration. We have a sum total of 6 CO2 molecules being produced, 4 ATP molecules being produced, 10 NADHs, and 2 FADH2s. Remember, FADH2s act in the same way as an NADH. They're an electron carrier molecule. All right, so this is what we have ended up with so far. Notice that by far the largest amount is in these electron carrier molecules. Right? We have produced quite a few of these. We've only produced 4 ATP. Right? So in this next step, we're going to focus on producing more ATP using these electron carrier molecules. All right, and that last step being oxidative phosphorylation. All right, so this is the final step of cellular respiration. This is where we're using our NADH and our FADH2 in order to generate large amounts of ATP. We're also going to see that while generating these large amounts of ATP, water is also going to be produced, right? So this reaction requires oxygen in order to produce water, and we'll talk about that process. But oxygen is required here. Right? This is why we need to breathe in oxygen. All right, so we're taking in, we're using those NADHs and FADHs too. Let's break this down. So I've shown you one picture on the screen of kind of the flow of what's happening here. But let's look at it piece by piece. So it's oxidative phosphorylation. Let's look at the oxidative part first. All right. All of those molecules of NADH, as well as FADH2, they are going to be oxidized at this point via the electron transport chain. All right, the electron transport chain is consisting of these protein complexes that are embedded within the membranes of the mitochondria. Right, so if you think about a mitochondrial molecule, if it's drawn out, it often is drawn with like an interior that looks very squiggly, right, because it's a bunch of membranes inside of the interior of the mitochondria. And now we're using those membranes as part of the electron transport chain here. We have many different protein complexes, right? They're numbered in this image, 1, 2, 3, and 4. I don't expect you to know what number does what thing here. Again, think big picture. But we have all of these protein complexes, and we see NADH and FADH2 are using those protein complexes. Something's happening here, right? We'll focus on NADH here, but the same thing happens at FADH2. But NADH is going to lose its electrons. It's going to give up its electrons that it's been carrying. It's also going to break apart and lose some of its protons. Protons being hydrogen ions, H+, right? Because if it's a hydrogen ion, the only thing that's left is a single proton. All right, so let's look at the electrons first, and then we'll look at the protons. So NADH has given up its electrons. Those electrons, so on this chart, it's kind of like these yellow stars here, are going to move through a series of reactions across these protein complexes. All right, so those electrons are moving through all of these protein complexes. Now these electrons, once they're done moving through the protein complexes, they have to go somewhere. Right? We can't just have free-floating electrons. They have to be captured by something. And what captures those electrons is the oxygen. Right? In fact, oxygen is the final electron acceptor. It takes in those electrons that just got done going through the electron transport chain. It also takes in a couple of protons, a couple of hydrogen ions, and creates water. Right? So oxygen is the final electron acceptor, and when it accepts those electrons, it produces water with the hydrogen ions. All right, so we focus on the electron. The electrons from the NADH, but also from the FADH2s. Right? They're being donated to the electron transport chain, and they're moving through a series of reactions across the chain. All right, now I'm going to flip the slide and take a drink of water quick. But now we're going to switch our attention from the electrons to the protons. All right, so the electrons have been stripped away from NADH and FADH2, but so have some protons. Notice that NADH becomes NAD+, right? And now we have some protons, some hydrogen ions, kind of hanging out on this side of the membrane. But they're not just hanging out, they're actually going to move, right? So these protons are actually going to move across the membrane using these protein complexes as transmembrane proteins, as carrier proteins, to go to the other side of our membrane. So by going to the other side of the membrane, now we're starting to create an electrochemical gradient. A gradient where on one side of the membrane, we have more protons or more hydrogen ions than the other side. All right, so these protons are moving. They are actually moving using the energy from those electrons. Right, so the electrons moving across these protein complexes, right, that movement of those electrons provides enough energy to help these hydrogen ions be pumped across this membrane. Right, and we can see that in a couple of these different protein complexes. The first one we see protons can move through. The third one, protons are moving through, as well as the fourth one. They're all pumping these hydrogen protons, these hydrogen ions, across the membrane, using the energy from the electrons as those electrons have moved. All right, so big picture here, electron transport chain. Electrons have been stripped off of NADH and FADH2. They have moved through the protein complexes. Oxygen has accepted them and water has been created. But that movement also allowed protons to move across the membrane and set up this gradient where there is more protons on one side of the membrane than the other. Right? This gradient is what is going to provide energy for the final part of oxidative phosphorylation. I'm going to pause, take a drink of water, make sure I'm not going too fast. All right, so we have this gradient. Let's see how we're going to utilize this gradient. All right, this is where the second half of oxidative phosphorylation comes into play. We are now going to focus on the phosphorylation part, right? So phosphorylation is simply a term that means adding a phosphate group onto a molecule. And if we're talking cellular respiration, we're going to be talking about phosphorylation in the terms of ATP to ADP, and then ADP to ATP. All right, so we have our molecule of ATP. We know that energy is held in the bonds of the phosphate groups, right? So when ATP is used, this phosphate group is going to break away, break that bond, release energy in order for the cell to harness and provide energy for whatever process it needs. Right? But when it loses the phosphate group, it turns into ADP, adenine diphosphate, meaning only 2 phosphate groups. All right? Well, this ADP needs to gain another phosphate in order to once again become ATP. It has to be phosphorylated. It needs to add a phosphate group to ADP. Right? Now, this is not this takes a lot of energy, right? Energy is needed in order to add a phosphate group onto ADP, right? So the phosphate group needs energy in order for ATP to become ATP, right? And that's the process that we're going to focus on for oxidative phosphorylation, is adding that phosphate group back onto ADP to become ATP. Now the ATP can once again be utilized by the cell. All right, so this is our last and our final step here. Remember that gradient we have set up of protons. We're going to harness the energy in this gradient and the movement of the protons across a membrane in order to provide energy to add a phosphate group back onto ADP. All right, and I'm actually going to run a little video up top here to help us visualize. Right, we have our protons. Our protons are on one side of the membrane in a higher concentration than the other. Now the only way for the protons to move to the other side is by utilizing a transmembrane protein. Remember, because the protons, they have a charge, right? In charged molecules, ions, they cannot simply diffuse across a membrane. They have to use a protein. And the protein being used in this instance is ATP synthase. Right? So the protons that are at a higher concentration on one side than the other are going to start moving through that protein, ATP synthase, to get to the other side. Because we're moving from areas to high to low now. We're moving with our gradient. But as those protons move through this ATP synthase, that movement is actually providing enough energy for ATP, or for ADP, to add on a phosphate group and become ATP. We can see ADP, it gets a phosphate, becomes ATP. So it's this movement of the protons through this enzyme that gives it enough energy to add that phosphate group back onto ADP, and now once again is ATP. All right, and there's a very specific term for this. We call this chemiosmosis. right? So chemiosmosis is the chemical synthesis of ATP through the movement of protons across a membrane or across a gradient. Right, big picture here is that the energy is coming from the movement of the protons through the ATP synthase. All right, and we think back to what made this possible. It was all those NADHs, all of those FADH2s that were produced in the first 3 steps. That has made this last step possible. And this last step produces ATP, but it produces a lot of ATP, right? In fact, at this stage, we produce from like 30 to 36 molecules of ATP. That's a huge number, right? So far, through the first 3 steps, we've only produced 4 ATP. And now in this last step, we're producing an additional 30 to 36. Right? That's a huge, huge number. That's great. Our cells need ATP. We want to produce a lot of ATP. Now notice it is a range. There's not a finite number here, right? And that is for a variety of reasons, right? Normally, not all of the NADHs are used in this process. They might be used in another process within the cell. Sometimes that gradient that is set up of protons is utilized by other parts of the mitochondria, right? So that we don't get the full amount of ATP produced through ATP synthase, right? But again, big picture, it's this huge number, right? Somewhere in the 30s. You'll see some textbooks probably saying like 30 to 34 or 32 to 38, all right? I think every textbook tends to differ on the specific numbers in that range, but they all agree that it's a large number of ATPs, typically in the 30s. All right, let's fill out this final part of our table here. And then I think I have one more slide and we'll move on to the content check. In the plasma membrane of the cell, which maybe they have to shuffle. So that means they just stay on the spot. Yes, you can say it.
Wait, what is regeneration?
Yeah, you're magically out of nowhere. Yes. They're hanging out, they're somewhere. All right, where does this stage occur? I hear whispers, but I can't hear the words. In the mitochondria, can we be more specific than that? In the membranes of the mitochondria, yeah. the inner mitochondrial membranes. This is where it's happening, across those membranes. All right, what is the end product of this stage? Yeah, I would say water, ATP. I think I also put in, I didn't put an ATP here because I put it in with the next line. But I also put in like NADH plus. That's what's left after it's given up its hydrogens and electrons. But also lots of ATP, right? Lots of ATP is also the end product here. How much ATP is being produced here? Yeah, like 30 to 36. I think my answer did 32 to 36. Somewhere in the 30s. Big ATP payoff. All right, I know it's a lot of numbers and letters, and you have to remember all of this. And I'm going to say repetition is going to be your friend here. Just draw it out many times, talk it out. Watch other videos so you help know the process and why this is occurring. I think videos are great here because this is a process, and videos show you things moving in real time, essentially, versus a slideshow that's just stagnant, one snapshot at a time. But repetition. Find a blank blackboard and draw it all out. I think that's going to be the most helpful way here. All right, my last slide is that I know we've talked about cellular respiration in terms of glucose, right? Because that is the most common molecule used in cellular respiration. And it's often the example molecule used in all of our examples, but it's not the only molecule that can be harnessed for energy, right? We can harness lipids, proteins for energy as well, right? They just have to go through a few extra steps before they enter into this cellular respiration process. And they might enter into the cellular respiration process at different stages, right? So this image kind of does a nice job of simplifying that. So for example, these fats, right, if we're looking at fatty acids, they're going to enter this stage as acetyl-CoA. And then from acetyl-CoA, they'll continue the cellular respiration, right? So there are other molecules that can be used to harness energy. It's not just glucose. All right, before we get to the content check, I want to tell you about the homework. All right, so this is a short unit. We really only have 4 lecture periods before the next exam, this being the second of the 4. So I want to make sure we stay on track. For most classes that meet Monday, Wednesday, Friday, they only lose 1 day of class because of Nobel. But us losing 1 day of class is half of our classes in a week. So I'm actually going to assign a video that I want you to watch before we come back on Thursday. So you have a whole week to do this. The video will be on fermentation and on overview of photosynthesis. And then when we come back on Thursday, it's actually one of my favorite class periods because we get to go over a bunch of different case studies applying our knowledge. Scientific literacy, just as a reminder, that is due next time we meet, as well as those exam points back opportunities. I do have a Nobel assignment, and I'm giving you an extra day for the Nobel assignment because I know there's a lot due on Thursday. But for the Nobel assignment, I have linked it on Moodle. I want you to go watch 2 Nobel talks, whether virtually or in person, I do not care. And then I'll ask you some questions about the talks themselves, right? Really, I encourage you to go to Nobel in person. It's such a unique experience to Gustavus to have the Nobel conference, to have a conference that doesn't just host ecologists or just host biologists, but has like historians and biologists and psychologists and a French chef all giving talks on the singular topic. It's really cool. All right, the content check. The part that is on Moodle, you should be able to finish now. That is due at the end of the day. This is the second part, content check number 3. Either write it down in a notebook and turn it in, or turn it in through Moodle before you leave today. But we're looking at a scenario. Right, our scenario is that there have been sailors adrift at sea. When they are finally rescued, they end up having this disease, suffering from a disease, and I'm going to butcher the name, called beriberi disease, right? And it's a disease where they have a severe deficiency of thiamine. And thiamine is an important molecule required for the activity of pyruvate dehydrogenase, which we did talk about. All right? So they have this deficiency in thiamine. They're also exhibiting symptoms where they have extreme muscle weakness, extreme fatigue, neurological issues, all issues associated with low levels of ATP. I want you to answer the 3 questions on the bottom before you leave today. And then that's all I have. And enjoy Nobel, because really, it's a cool conference.